Pathway-based human disease clustering tool using self-organizing maps by James Andrew Sarmiento, Geoffrey Solano, and ANGELYN. The exploration of disease-to-disease associations by using a system-level biological data is one of the greatest challenges in modern biology, but it is expected to yield new breakthroughs and insights in the medical field. There have been many efforts to create publicly available databases for future researchers planning to discover new knowledge and test hypotheses. One example is the Kyoto Encyclopedia of Genes and Genomes, or also known as KEG. In order to discover underlying patterns in a large dataset such as this, a data mining and knowledge discovery technique should be used that enables data compression, visualization, clustering, while still preserving the topological properties of the data. In this study, an unsupervised artificial neural network, self-organizing maps, is implemented as it is capable of doing the said requirements, making full use of the publicly available pathway databases. Additionally, other data mining techniques are utilized such as hierarchical and agglomerative clustering for other visualizations. With the establishment of pathway databases containing perturbed molecular networks or pathways of these diseases, such as KEG, it opens opportunities for future studies to use data mining techniques to discover knowledge of disease similarities that could potentially lead to new breakthroughs. There had been related works with regards to the topic of disease similarity. A study of Yang produced DNetDB, which is a tool that generates network of diseases by comparison of regulation of gene expressions. Ping used combination of common susceptibility genes and HPPIN to study shared pathways, neurodegenerative diseases, and Turkmani used pathway analysis on 7 common diseases by GWAS, producing a table of Pearson correlation of coefficients from these common diseases. Self-organizing maps has been used by many studies for finding relationships and classifications. Chavez Alvarez used SOM in discovering possible gene relationships to DNA microarray databases, while Al-Ajlan tries to classify pathway analysis techniques and the data mining approaches that can be applied in pathway analysis. What is a pathway? Pathway is defined as a collection of genes that serves a particular function and/or genes that interact with other genes in a known biological process. This is a pathway of allograft rejection from KEG. Self-organizing maps is a neural network that represents a high-dimensional data as a quantized two-dimensional image in an orderly fashion. It maps data items into a point or node in the map, preserving distances of these input vectors in the map. These units are called neurons that form a two-dimensional lattice in which the data items are mapped into, preserving topological structure of the data. The input vectors are called the input layer while the neurons are called the output layer. This image shows the mapping of the input vector into the corresponding neuron in the self-organizing map. Other visualizations that can be created from SOM are U-matrix and component planes. U-matrix shows distances between neurons by using gray colors, where darker coloring means larger distance, serving as cluster separators, while component planes, which functions like heat map, indicates presence and absence of the target variable. The tool also included two clustering techniques. Hierarchical clustering is a method of cluster analysis seeking to build hierarchy of clusters, while K-means algorithm places cluster centers which converges to a local minimum of the objective function as the sum of the squared Euclidean distances between data points and the nearest cluster centers. System design and results. Here is the use case diagram of the tool. It provides three main functionalities. One is start clustering for the training of SOM and analysis of diseases. Second is uploading KGML files to the system's database. And third is viewing the user guide. The data flow diagram is shown here. The user uploads the KegXML to the system and creates a disease entry which is stored in the database. To train SOM, the user provides the input parameters and chooses the disease from the list. To visualize the data, the user provides configurations to the visualizations provided and is given the visualizations. Lastly, the user could view the tutorial. To use the main functionality of the tool, the user opens the application and goes to the Visualize panel. The user selects the diseases to be included in the pathway, entities or components to be used for the analysis, which are entries such as genes, proteins, and enzymes, relations, which are relationships or interactions of two entries, and maps, which are pathways on their own. The user goes to the next step to configure the map configuration. The user could set up to 3 different self-organizing map settings, and the tool automatically chooses the best among all of the runs. Other visualization modules are now available to the user. First is the SOM lattice itself. 30 diseases were included, and maps is the option selected. From the result that it is in the paper, it could be observed that substance addiction such as morphine addiction, amphetamine, alcoholism are projected near to each other. Cancer diseases were also relatively near to each other. From the U matrix with neighborhood size of 3, it gives a rough estimate of how the data is naturally clustered into groups. Light-colored nodes, like in the group of substance addiction and the cancer diseases, indicate that they tend to form their own clusters, while the black bordering nodes indicate possible distant values from the rest. From this, we choose k equals to 5 for the k-means clustering. The k-means cluster tried to group the data into 5 clusters. As previously seen, substance addiction and cancer diseases form their own respective clusters. Interestingly, diseases that are not obviously related form their own clusters too, such as tuberculosis, hepatitis A and B. Alzheimer's disease, type 2 diabetes, and other form their own clusters too. The k-means visualization also gives a summary of the cluster, including the common pathway entities per cluster. Component planes give a heatmap functionality to the user where it can project the component planes to the clusters and check the distribution of that pathway entity in the map. The tool also provides another clustering technique and another perspective of the cluster hierarchies of the diseases. From here, the previous results could be observed as the substance addictions and cancer diseases form their hierarchies of clusters. The cluster of tuberculosis and hepatitis are also evident. PathSOM is a tool that allows different visualizations of the relationships of human diseases retrieved from the Kyoto Encyclopedia of Genes and Genomes, also known as KEG. The visualizations generated by the tool could be used for future studies that will tackle relationships and risk factors of smaller group of diseases and help provide breakthrough insights in the study of diseases.